An assignment about genetic screens. And genetic screens are something that scientists do in a laboratory, and they involve taking a population of organisms and exposing them to a mutagen, like X-rays, for example, and that mutagen is going to increase the rate of mutagenesis, and it's going to create what we call induced mutations because they're done in the lab, exposed to a mutagen, and but they're still random. And so this population of organisms, and the numbers are like 10,000 of the organisms, are mutagenized, and of course it's only done in simple model organisms like a nematode, a fruit fly, a zebrafish, because we need to have lots. We need to screen a lot because the mutations are random, and so there are random mutations in every gene. So a given organism might have one or two, a small number of new mutations, and we have to screen them to see which ones have gotten a mutation in a gene that's important for what we study. Most of the topics in our course started out by geneticists doing a genetic screen to start finding the genes that were required for the function. So the assignment is going to be to look at and imagine early studies of muscle differentiation, because we know those genes. And but the example I'll talk through in this video is going to be the scientists who studied the nematodes that were undergoing programmed cell death or apoptosis. And so the idea is, the assignment is for you just to learn about the nematode and then think, how would this be equivalent in the early studies of the muscle differentiation? And it would have been done in a simple model organism like fruit flies. So take 10,000 fruit flies, or 20 or 40,000 fruit flies like that, and mutagenize them, expose them to a mutagen in a lab, random mutations. So we should start looking at them, and we look for, in the case of the assignment, a mutation that doesn't have muscles, that has some defect in muscle development. And the interpretation is, well, that animal, which we call a strain, because we may breed it and maintain that mutant in the animal, so we can study it. That mutant strain must have a mutation in a gene that's necessary for muscle differentiation, that's required for muscle differentiation. So here, let's just read the assignment. So we got to have it due by either email or handed in at the start of class on Tuesday, the 24th. So draw labeled pictures, and so just simple labeled pictures to get that visual and that spread out learning, and write explanation for how to use complementation, and we'll talk what that is, to determine the results of a genetic screen. So that's one of the terms we want to learn here. And so for this assignment, 8 loss of function mutants were isolated in the screen. The names of the 8 strains can just be 1 through 8. Assume 2 of the strains have mutations in the MyoD gene, right? We're just beginning to learn about it, but we have the knowledge of modern knowledge of knowing that this is an essential gene. And so let's say we isolated it, not surprising, but really don't know its name. So tentatively, we're just going to call our mutant strains 1 through 8. And we'll describe what that means in the video in the context of the nematode programmed cell death genetic screens. And then another strain has a mutation in the myogenin, we know that. And then 5 of the strains have mutations in MIF5. We haven't talked about MIF5, you don't need to learn about it. We're just using it here. It's another gene that's involved, like myogenin and MyoD, in muscle differentiation. Okay, so that's the assignment, and you can be thinking about it as you learn this, and we really just repeat the talk that I'm going to do about the nematode and programmed cell death with this idea that, what if you did the screen for something else? Because that's an important concept, is, oh, well, we learned the genetic screen about one story, and then we could apply it to others. And again, most of the stories that we tell in our course started out early, like back in the 1990s, as genetic screens, where we're like, we have no idea what genes are involved in programmed cell death. We have no idea what genes are involved in muscle differentiation. So we got to find mutants that that have defects in programmed cell death or muscle differentiation, and then learn what are those mutants and how do they work. Not just genetically, but how do they work biochemically? What are they doing? So here's just a term, and the term is loss of function, and that's a term that's going to be defined toward the end of this video, and I just want to highlight that because you'd be like, well, what's loss of function? And the assignment is explained as we go through this video. So a loss of function allele is just to start out to define it. It's a mutant allele that doesn't function as well as the normal wild type allele. So wild type alleles are considered to be just a normal, completely normal functioning allele. And the term is explained near the end of this video. This is the most common mutant allele to find because random mutation is most likely to damage a gene's function. A null allele, which we already know about, is a special example of a loss of function allele because a null allele we know has no activity, has like 0% activity. So that's just one of a type of loss of function allele. And these would be true for any gene. It's a general genetics term for any gene. We can have a loss of function of myogenin. That means it just doesn't work as well as the normal myogenin gene. We can have a null allele, a myogenin. It doesn't work at all. It's a special loss of function. And I just wanted to point that out here because here it is in the context of the assignment. You might be like, oh my gosh, I don't know what that term is. And there are other terms in here that will get introduced as we go forward. So what you'll do is you'll come back to the assignment, but I want to show it to you at the front end so you can be paying attention to what the expectation is, and that is we're going to learn the core of this and then we're going to apply it to this muscle example. Okay, so it comes from the first programmed cell death story, and it goes like this. So we can use those DIC optics in a microscope to look at living nematodes, and we see that the nematode life cycle looks like this. We start out with a fertilized egg, and then the cells divide to make an embryo, and then the embryo hatches and it starts moving around, crawling around, and it goes through 4 larval periods, L1 through L4, the L1 larval stage, to adulthood. So that's its life cycle, that's its timeline of developing. And oh, there's a slide about the DIC, so we can see it. And John Sulston, again, we don't need to remember his name, but he must have gotten very tired because this shows from left to right this timeline of that development. So these would be cell divisions that happen just in the L4 as it becomes the adult. So you can see that some of these cell divisions are stage specific because over here is the fertilized egg, and it makes a first cell division, 2 cells, and those are going to make all the cells in the embryo. They divide, and some of these are to make the embryo, then the L1, L2, L3, L4, and the adult. So there's a specific pattern of cell divisions that have the same timing in all nematodes and also the same fates or the same outcomes, and that's said to be an invariant cell lineage, and that's kind of unique to this nematode. It's probably true for other nematodes. I have no idea, and people don't know either because the scientists really focused on this organism and just watched it develop and mapped it out. And here's just an example of that. So the fertilized egg, and it divides in 2. So that's shown here. It divides in 2, and we've just zoomed in on that. And Sulston named these because you could see him. He named them, and he could name them as being different because, for example, this P1 is going to be the cell that gives rise eventually, so these are further cell divisions, it gives rise eventually to the entire germline, and a germline is cells that could make sperm or egg. So that's a general term in animals, is that sperm and egg are called are from the germline, and they're called gametes. Sorry, just forgot that term. So they're called gametes, the general term, and they're from the germline. Germline cells are cells that can make gametes, either sperm or egg. And so, see, there's a distinct fate outcome for these cells in that, yeah, they both can produce some specific muscles, but it's different muscles in the body, and they make other things that are distinctive. Okay, great. So that's the idea. And then if we zoom in on the embryo and look just at the embryo and not at the after hatching, so just before hatching, you see that there are cells that die in the embryo. And these are going cells that died through programmed cell death. These are just cells that stop dividing, and they didn't die. They live for the life of the organism. But some die very early before it hatches. So this is a great place to do a genetic screen, because you could just do a mutation, an induced mutation, and then just look for examples of embryos where these don't die. If they don't die, there's a problem with programmed cell death, and you've isolated a mutant allele, and you can start breeding that strain, and you can breed it because easily in this case, because these nematodes are hermaphrodites, the organism can produce both eggs and sperm, so it's pretty easy to breed it and maintain a strain, and the strain would have one specific mutant allele. And you can maintain that strain and find out, well, what about that allele? So this is a big experiment in a research lab, and it says identify mutant nematodes by inducing random mutations in lab and then doing a genetic screen. So those random mutations in the lab are called induced mutations, and that's different than the spontaneous mutations that are randomly found in nature. The genetic screen involves looking at each nematode in the mutated population. Again, the population is tens of thousands of animals, so it's just a day after day after day of looking under a microscope, and you're looking for an animal that doesn't have programmed cell death. It has a problem, a defect, a loss of programmed cell death, and so it's really, initially the genetic screens are very labor intensive, and but as scientists learn more and more about the programmed cell death, there are ways to make the screens easier, and we'll probably talk about that some in class. So saving those phenotypes with defective programmed cell death. Each mutant has a mutation in a gene required for programmed cell death. Importantly, we don't know if it's in a gene we have already maybe studied, or if it's in the same gene as other strains. So we need to study them further, or just isolate them. Each PCD mutant is bred and maintained is called a strain. So we isolate as many different mutants and study each strain. And so this is called a genetic analysis, and just maybe it's a little bit out of order with the other ones, but where it's a search for new mutations to study genes, a discovery of genes that are required for function like programmed cell death, or in the assignment, also differentiation. So you could find a mutant in nature, that's spontaneous mutation, or you could do a genetic screen and find induced mutations. Induced mutations by exposing the organisms to a mutagen like X-rays. It just increases the random rate of mutagenesis. We'll find more mutants than if we just walked through nature. Then screen the organism for individuals with defects in the phenotype you want to study. Then determine which gene was mutated in each organism that caused the mutant phenotype. So that's a genetic analysis. And again, we could do it in any species, but it's just really impractical to say to do it in mice, right? Mice is a common model organism, but you can't grow 20,000 mice and keep them all separate and organize them in all different cages like that. So it can only be done in these really simple model organisms where you can grow tens of thousands, hundreds of thousands of fruit flies and study them like that. Okay, so let's say that we do a mutant screen and we identified 6 mutant strains, and we just tentatively label this. We have no idea what genes the mutations are in. So this is a strain, and it's one we just found, and then we bred it and maintain it, and separate from all the others. So we have a pure strain with that mutant allele. Then on another day we found another mutant strain. Okay, now these are said to have independent mutations. Right? They're just different animals in the population who got hit by an X-ray, and one got a mutation, and they're random. So the chances are that these would be in different genes. They're independent. They could be in different genes that are both required for mutagenesis. And that's great because we're trying to find all the genes that are required for programmed cell death. There are probably several. So let's try to find them all. Now, these either have mutations that are in 2 different genes. That's great, because then we can study each one, and this would be maybe MyoD, and this would be maybe myogenin, right? 2 different genes. In this case, they would be programmed cell death genes, and we don't know the names of them. That's fine, but that's the concept. On the other hand, maybe these are two different alleles of the same gene. We have 6 mutant strains, and maybe there's only one gene, and that's not true, but if there was just one gene involved in programmed cell death, well then everything we isolate would be different mutations in the same gene. Different alleles, and representing different alleles of the same gene. Different alleles because they are independent mutations. They're really unlikely to be the same mutation. Okay? So we want to find out, what did we discover? Because we start genetically, and we're just going to go through this as an example of what geneticists do during a genetic screen. They isolate these mutants, they maintain them as a strain, and then they find out, well, how many genes have we studied? How many have we identified? Like that, okay? And so here's what they do. They take this a mutant, and they breed it with that mutant, and they look at the babies of that cross, that breeding experiment, and this is called a complementation cross. So we have to draw this out and just explain this, and maybe it'd be best to do this over here. So let's say, let's represent strain A, and again, this is general. We're focused on programmed cell death, but it would be true in any genetic screen. And here's a gene, we'll just draw it like that. And the organism always has 2 copies of the gene, one from mom and one from dad. And this is the gene that has a mutation. So let's just draw it like that. There's a mutation in the gene. Okay? Now, strain B, let's make this maybe, let's make it the same color. Okay, so strain B. So let's say that strain B has mutation in the same gene. Okay, but a different place. It's random. It's just a different allele, but both of them are defective. And when we do this cross, no matter what, the babies will have 2 alleles, one from mom, one from dad, and one of them is defective and the other is defective. So they have a defect. They have the mutant phenotype. In this case, the cells fail to die. In the assignment, the cells fail to form muscles. And they're really different mutations, and they're independently isolated, but they're in the same gene, so they're different loss of function alleles in the same gene, and we see the mutant phenotype. There's a problem. And we say for this mutation, they fail to complement. So that's the complementation cross term. They fail to complement. That means they fail to kind of cross each other out, and we'll describe that in the next image like that. So they fail to complement. We see the phenotype, and these are we could say just alleles of the same gene. We've isolated 2 alleles. Strain A has a mutation in the same gene as strain B, and it's an allele of this gene, like that. Okay, so that's really the same gene. Now, here's the other possibility. Just redraw this again. Okay, the other possibility is this, is that they're in a different gene. So strain A and strain B. And here's strain A has a mutation like this. And the mutation is here. Now, that's a gene that's essential for programmed cell death. Let's say there's a second gene that's also required for programmed cell death, or whatever it is we're studying. Now, it doesn't matter that this gene is fine. This is wild type, one from mom, one from dad. It doesn't matter because this gene, I should put another one in there, this gene is defective. So, if we look at its offspring, just crossing it to itself, the offspring would be defective. Now, let's say that strain B picked up a random induced mutation and happens to be in a different gene, a different gene that obviously is also required for programmed cell death, because that was our screen. We're looking for mutants that had defects. So these both have defects in programmed cell death, but it's due to a mutation in a different gene. Okay? So now let's look and see what happens when we do this cross. In this case, when we do this cross, right, what we'll see is one of the genes is from strain A and has the mutation. The other comes from strain B and it doesn't have the mutation. So this has one defective and one normal or wild type allele, like that. And then over here, we see the same kind of thing happening, where it got a normal allele from mom and dad, and mom or dad of strain A, and a mutant allele of strain B. So this one also is, in this case, they're both heterozygous, and they have a normal and defective allele. There's a good chance when we look at this that the that this offspring will be normal, because it has a good version of each of the genes, and there's a good chance that it'll be normal. Maybe it'll have a little problem, but maybe it'll just be totally normal. We have to do the test and see. And if that's the case, if it's if the outcome is normal, we interpret that as this, where one strain complemented or rescued is another term. So these two crosses, they complement. And or another term that scientists use sometimes is they rescue the mutant phenotype. They rescue, complement the mutant phenotype. And we interpret that to be if two strains complement, the mutations are in different genes. And we like that because now we found two different genes. What's that gene do? We go on to study it in genetics and biochemistry and molecular genetics and so forth. What does that strain do? Okay, so that's the idea of a complementation test cross, is we do crosses to determine which of these are from the same gene and which are different genes. Okay, so we have to do all these crosses. So that's another example of genetics in a lab. It's a lot of work. Lab genetics is a lot. Traditional lab genetics is a lot of work, a lot of breeding experiments, because that's what genetics starts out with. So let's say that we do all of the crosses. So again, cross this one with that. Do they complement or not? Then cross that one with the others. Do all the possible combination crosses and ask, do they complement or do they fail to complement? If they complement, they must be mutants, alleles in two different genes. If they complement, if they fail to complement, they are the same gene but different alleles. Okay, so let's say these two fail to complement, so they represent different alleles of the same gene, and these are not that same gene. These crossed with all these complemented, so these must be represent a different gene or genes. Then we found these two failed to complement. They're different alleles of the same gene. So this is, we could just call it sort of tentatively gene 1. This is gene 2. And we've got two alleles of gene 1, two alleles of gene 2. And what about these guys? All the other crosses complement. How many PC genes have we discovered? And the answer is, well, we have two alleles of gene 1, two alleles of gene 2, one allele of what must be a gene 3, and one allele of what would be a gene 4. When you do your assignment, one of these genes you can label as MyoD, just to help you kind of get a feeling for it, one as myogenin, and one as MIF5. We'll be looking at 3 different genes in that screen. The number of strains and their complementation crisscross are different this, but just to show you the idea of the assignment. So here for this PCD thing, we've discovered 4 genes, and we go on to study them. There are 4 different genes. We have some alleles. We isolated 6 strains, and out of it we got 4 genes. That's awesome. It's random, so we could do a screen, and it could be that there are 4 genes involved in programmed cell death, and maybe we would just get 6 strains all of gene 1, just by chance. So we have to do a lot of screening. Lots of times we go through a screen, we screen 20,000 animals, we get the genes a set of genes, and then we go through another round of 30,000 to find them. We're trying to find all the genes that could be involved in programmed cell death, and it takes a lot of screens because they're random. The mutations are random. So this is just from our book, and it just shows that in the nematode, there are at least 4 genes involved in programmed cell death. And so this just shows like proteins of 4 genes, the protein products of 4 genes, and we don't need to learn their names, but there are the final names that the geneticists gave them, like that. And we don't need to learn these names, but just to give you a vision of this. And you can see that they have this activity, like that. Just as a side note, you can see this is an activator of programmed cell death. So if you had a decrease in that allele, you would have a decrease in programmed cell death. This one is an activator of that one. So this one also is required for programmed cell death. If you lost it, you would have decreased in programmed cell death. Now look at this though, because this one, see that little bar there, this means it represses or turns off that one. So just to show you, our story is more complicated because some genes repress programmed cell death, and if they got mutated, you wouldn't see a loss of programmed cell death, you'd see an increase in programmed cell death. The cells would die earlier, or additional cells would die that didn't normally die. So genetics is complicated. It's understandable, but it's got a lot of stuff to it, right? There could be protein made from a gene, SED9. SED9 is required for programmed cell death, but it's repressing it. And then look at this eagle 1. It's a repressor of a repressor. That means it's really an activator because it turns off the repressor. So this one would come out of our screen. This one would come out of our screen because we're just looking for a failure for cells that go through programmed cell death. This one would not come out of our screen, right? So the nematode geneticists had to work with that, and it's like a murder mystery novel, where you just have to work out all the evidence and draw a map out like this. Okay, and just to show you, scientists did this at work initially, and other scientists in fruit flies and other scientists in mammals like mice eventually used these discoveries to find similar genes involved in programmed cell death. You look at all the activation and repression that occurs like that, and we'll talk about this in class, but these are orthologs like that. So the matching colors are orthologs between these species, and you can see that programmed cell death has been conserved, has maintained really similar genes over evolutionary time, because the last common ancestor between all these must have already had these genes involved in programmed cell death, important for development of them. So what we're going to do is we're going to learn these names, BCL2 and caspase-9, and not these other names from these model organisms, because they aren't the common modern terms used now for these genes. Okay, so again, back to this, these two failed to complement, so they're different alleles of the same gene. And you might think that a geneticist is really happy when genes complement, because that means when they complement, they're studying a different gene, and they want to find all the genes. So when they find 2 genes like this and this that fail to complement, they go to the Red Sox game because they found 2 genes. And remember, in our example, they found 4 genes. That's great. Now, just want to work through some terms, because they're also excited for different reasons when they find different alleles of the same gene, because the different alleles of the same gene give them powerful strategies in genetics to study this gene in detail. And so we're just going to go through some terms where were terms of the alleles. And so these would be the alleles that would be in the population in the original screen, or these would be the type of alleles that would just be found out in nature through spontaneous mutation. So let's say that we isolated initially 11 mutant strains, and we did the complementation test crosses, and we grouped them, and we found that of these 11 strains, how many is that? 7 of them were actually all in the same gene. Independent mutations in the same gene. They're probably not the same mutation, because that would just be so unlikely to have exactly the same mutation when you're doing random mutations. And so they're probably in different spots of the same gene. And they all cause a decrease in activity. So one might be here, one here, another one here, like that, stuff like that, right? Okay? And so what is valuable at that? To understand that, just kind of introduce that, we're going to introduce these terms that are important terms that we want to learn about, that are from genetics, and include that term loss of function. And we're going to introduce that because our book uses that loss of function when it talks about the second story that we're going to learn about. The second story we're going to learn about, talk about in class, is RNA interference. And to understand RNA interference, they didn't even know it existed. Scientists were doing genetic crosses, and they found genes, and they found a gene that was involved in RNA interference, and they were like, what is that gene doing? Okay, oops. So let me go over to this and talk about this. So here's something that is a nice visual that you'll never see drawn. And it goes like this. And the reason you won't see it drawn is because it's a real thing. It's true, but it's hard or almost impossible to make these measurements. And so what we're going to do is we're going to look at really kind of it's kind of like a graph or a table, and this has the gene activity. And we could be looking at any gene. We could be looking at that SED9, that programmed cell death gene, or we could be looking at myogenin, or we could look at hemoglobin. So it's any gene, and this is the gene's activity. So a higher and lower activity. And it's really kind of impossible to really, truly quantify the activity of a gene. It's true that each allele of a gene has an activity, but you really can't quantify it. So you don't see this drawn out, but it's a nice visual for us to think about. So here is an allele, so these would be alleles, a version of the gene that just has 100% activity. And that's what we talk about when we say wild type. The idea of wild type is it's just a typical, normal, good gene. And the implication is it's just functioning normally, it's functioning at 100%. And now in our mutant screen, all the alleles we discover map on this spectrum. So remember, we found A, B, and I think it's G, H, I, J, K. Those are our strains that we found, and we complementation crossed them, and they all failed to complement. They're all in this different alleles of the same gene. So where do they map on here? And the answer is, this is a decrease in gene function. This is an increase in gene function. And because we do random mutations, almost all these genes, alleles, are likely to map here with a decreased function. One or more of them might have 0%. There are a variety of ways we make a null allele, so let's just say that A, B, and K are all null alleles. One way or the other, we just happen to completely destroy the gene function there. So that's an example of this. And they're all likely to be a decrease in function. So let's just put them on here like this, and we'll say that we have like G and I and J and H, like that. Okay, so that's our 7 alleles. They're all loss of function. And so we got to have some terms from genetics to help explain this. So alleles, like all of ours, that have a decreased function are said to be loss of function. the loss of function includes the null allele, but it's any allele that has a decrease. So loss of function is a little bit of a slippery term because it sounds a little bit like it might be null, but it means decrease of function. And there's another term that's synonymous from genetics, and these are said to be hypomorphs or hypomorphic alleles. And so these are terms that we need to learn because even though we won't talk about this very much in our course, this is kind of the heart and soul of the origin of a lot of modern developmental biology. Is let's find the mutants that are in the genes that are required for this developmental process, program cell death, muscle differentiation, and so forth. These alleles all have, and again, we can't measure it, but just to give you the idea, maybe this one is knocked down to 94% activity, and this one is 73, and this one is 52. And they have a loss of function, they have a defect that's observable, and it's 21, like that. So they're all a loss of function, they can be different, like that, and they are a decrease in function. Those are the most likely ones we'll identify. And while we're here, we just want to also add some terms because you'll see we'll want to use these terms. So again, the book uses this loss of function term in the textbook. And it doesn't use that very much, but we want to have a feeling for that term as we go through and can use it. And we won't go into detailed examples like this again, but we want to have visible, so I can hold up my hand and say decrease of function, hypomorph, loss of function, allele. Okay? Here, we have this curious one, an increase in function. And to start out with, you think, oh yeah, like 120%. And that's surprising. But imagine that you could have a mutation in the hemoglobin gene, say, for example, and it would just be better at getting oxygen from the lungs to the tissue, right? So that could be an increase activity like that. And an increase of activity is called a gain of function mutation or a hypermorph. And those are rare because how could you mutate a gene to make it like better or different in an improved way? It has its regular wild type function and it can do more. And we'll talk about these because these are rare, very rare, and we likely would not find them in our screen because we're looking for loss of function, right? And it would be pointless to do a screen looking for gain of function, kind of, because they're so rare. But they can be found as spontaneous mutations in nature, and they are the source of lots of evolutionary change. So that's a really important concept in genetics, is that we usually think and study a loss of function, decreased function, and you're like, that's not going to help the organism survive natural selection, survival of the fittest. But there are these guys that could. And our initial story, just for this video to get the term, is they somehow have an increase in function, like that. But it isn't saying that here. It's saying a gain of function. So it's like an increase or an additional new function. Okay? So we want to get those terms out of there because, again, our book uses loss of function in the second story, talking about RNA interference, and it talks about gain of function, and these would be alleles. So we talk about a loss of function allele, and we're saying like, oh, could be a null allele or any decrease in gene activity. Then we talk about a gain of function or hypomorph. We talk about some kind of a new gain additional function, not a loss of function, but a gain of function. And so we're going to stop the video here. I'll scroll back to the assignment, because the assignment is just to draw out, I'll show you this, draw out this chart, and instead of saying A, B, C, D, E, F, say there are going to be 8 new strains, 1 through 8, do the complementation crisscross, and you get, you show this, where's the assignment? You get 8, and then you line them up like that, and then you draw out, well, which one eventually becomes myogenin and myod, because we have 1 strain, 1 strain of that, and then you just explain briefly what you find when you do the complementation test cross. You don't need to explain, the loss of function terms. You can just understand that they're likely to be loss of function, and you just need to explain that briefly. So now what you do for your assignment is not very much work. Draw that out. Draw this out with a 1 through 8 and show which of these complement and which are eventually going to be discovered to be MyoD, myogenin, and MIF5, just to get a feeling for how a complementation test cross and a genetic screen can be done to study any phenotype, programmed cell death, muscle, so forth. Insulin, insulin production and activity like that. Anything that has a mutant phenotype that we can observe and that we can study in a simple organism that we can grow thousands and thousands of. Okay, so the actual activity of your assignment is just short. You don't need to outline this whole video. You don't have to say anything about loss of function in the video and map that last piece out. That's a separate piece. So it's a pretty short assignment that you can get done, but you do have to walk through this whole video to get to it. Okay, great. And so that's it.